6 miles below the Pacific Ocean, in total darkness, under pressure 1,000 times greater than what you feel right now, scientists just found something alive. Thousands of them. Worms, mollusks, creatures eating toxic chemicals for breakfast. Creatures that rewrote what biology says is possible. We're going to meet a fish with no skull, an octopus that flies, a worm with no mouth that drills through bone, and a single cell the size of a dinner plate. We'll find out why your grocery bag is sitting at the bottom of the world and what scientists think this all means for life on other planets. If you enjoy this journey, subscribe and hit like, and prepare yourself. We begin. Picture the tallest thing you've ever seen. Now bury it. Mount Everest stands about 5.5 miles above sea level. Scientists used to say that if you could somehow pick it up and drop it into the deepest part of the ocean, the whole mountain would disappear underwater and you would still have more than a mile of dark ocean sitting above its peak. That deepest part is called the Mariana Trench. It sits in the western Pacific Ocean, roughly 300 miles southwest of the island of Guam. The trench is shaped like a crescent, about 1,500 miles long and 40 miles wide. At its deepest point, a slot called Challenger Deep, the ocean floor drops to roughly 7 miles below the surface. 7 miles, straight down. To understand what that distance actually means, imagine standing in New York City and looking toward Los Angeles. That drive is about 2,800 miles. The Mariana Trench is about a quarter of that distance, except it goes straight into the earth beneath the ocean. For most of human history, scientists assumed this place was empty, dead. The pressure alone, they said, made life impossible. The deeper you go in the ocean, the more water sits above you, and all that water pushes down. By the time you reach the bottom of Challenger Deep, the pressure is over 1,000 times greater than the air pressing against your skin right now. That is enough force to crush a steel submarine like an empty soda can. The temperature down there stays near freezing, usually between 32 and 36 degrees Fahrenheit. Zero sunlight reaches anywhere close to the bottom. The nearest photosynthesis, the process most life on Earth depends on, stops within the first 600 feet of the ocean. Below that, there is no energy from the sun, no warmth, no light. In the 1900s, a British expedition dragged nets along the seafloor in different parts of the deep ocean and found almost nothing. Scientists built a theory around that idea. The deep ocean is a desert, maybe some tough microbes, maybe a few scavengers, but complex animal life, communities, ecosystems, the pressure would tear them apart. Then something happened that changed everything. In the early 2010s, researchers started dropping cameras and traps into the Hadal Zone, the scientific name for ocean regions deeper than about 4 miles. The word Hadal comes from Hades, the ancient Greek underworld. Fitting. Because what those cameras sent back looked like a world that had no business existing. They found fish. They found shrimp-like creatures swarming by the thousands. They found worms. They found creatures building entire lives in conditions that, by every rule science had written, should have killed them instantly. And in the last few years, researchers took it further. A submersible dove into some of the deepest trenches in the Northwest Pacific repeated the dives more than 20 times and discovered what scientists would later call the deepest and most extensive communities of chemical-eating life ever recorded on Earth. Thousands of animals, 6 miles down, thriving. Biology had been wrong, spectacularly wrong. The ocean did not become empty as you went deeper. It became stranger, and stranger is exactly where we're headed. Because the fish scientists found down there has a body that should be physically impossible. Stand outside right now. The atmosphere pressing down on your body equals about 14 pounds of force on every square inch of your skin. You do not feel it because your body grew up with it. Every cell, every organ, every bone was built to handle exactly that load. Take that pressure away and your blood would start to boil. Add too much more and your cells would collapse. At the bottom of the Mariana Trench, the pressure reaches over 16,000 pounds per square inch. That is more than 1,000 times what you feel standing outside. It is the equivalent of having about 50 jumbo jets stacked on top of a single square inch of your body. Here is a classic experiment that shows exactly what that pressure does. Scientists have sent styrofoam coffee cups down to the bottom of the trench on the outside of research vehicles. A styrofoam cup before the dive is about the size of your fist. When it comes back up from the deepest depths, it is the size of a thimble. The pressure squeezed out every single air pocket inside the foam, compressing it down to almost nothing. The cup still exists, still holds its shape, but it is tiny, dense, crushed from every direction simultaneously. That is what pressure does to air-filled spaces, pops them, flattens them. This creates a serious problem for any animal with a gas-filled space inside its body. Humans have lungs. Fish, in shallow water, have swim bladders, a gas-filled sac they use like a built-in life jacket to stay buoyant. At extreme depth, a normal swim bladder would implode. The gas inside would compress to almost nothing, and the fish would lose all control of its position in the water. The pressure also attacks molecules themselves. Proteins are the tiny machines that run every cell in your body, controlling everything from muscle movement to digestion. At high pressure, proteins start to warp. They lose their shape. When a protein loses its shape, it stops working. Think of a key that gets bent. It no longer opens the lock. That is what pressure does to proteins in a normal body at 6 miles deep. And it gets more specific than that. The membranes that surround every cell, the thin walls that hold your cells together, harden under extreme pressure. A hardened membrane cannot let nutrients in or waste out. The cell dies. So how does anything survive down there at all? The answer turns out to involve a molecule scientists call a piezolite, a chemical that cells pack inside themselves specifically to counteract pressure. It works almost like a cushion wrapped around each protein. Even as the water outside pushes in from every direction, the piezolite keeps the protein from bending out of shape. It holds the biological machinery together when the physics of the environment are trying to tear it apart. Deep-sea creatures do not just tolerate pressure, they have evolved chemistry that uses the pressure, leans into it, works alongside it. And one animal does this better than anything else scientists have found. A small, pale, ghostly fish discovered hiding in the darkest place on Earth. Its bones are soft, its skull is open, its skin is transparent, and it rules the deepest waters on the planet. In the summer of 2024, a team of scientists lowered a submersible called Fendouge into the water above the Western Pacific. Fendouge means striver in Mandarin. The name fits. The craft would need to work hard. Over the course of the research expedition, it dove into some of the deepest trenches in the Northwest Pacific more than 20 times, carrying researchers inside a pressure-hardened sphere the size of a large bathroom. Each dive took hours just to reach the bottom. What the team saw on those dives did not match any expectation. 6 miles down in 2 separate trenches, the cameras picked up something that biologists had theorized for years, but never actually seen at this scale. Thriving, dense communities of animals building entire lives from chemical energy alone. The seafloor was not a wasteland. It was busy. Thousands of tube worms clustered in mats across the sediment. Clams gathered near cracks in the rock. Smaller creatures moved between them. The whole community was organized around seeps, places where fluids rich in methane and hydrogen sulfide bubbled up from inside the earth through fault lines in the seafloor. Methane, hydrogen sulfide. These are chemicals that would kill most creatures within minutes. Down here they were the menu. The scientists published their findings in the journal Nature. They called it the deepest chemosynthesis-based community ever documented on Earth. Chemosynthesis is the process these organisms use instead of photosynthesis. Where plants near the surface grab energy from sunlight, these creatures grab energy from chemical reactions involving toxic gas. That finding alone would have been enough to rewrite textbooks. But the implications went further. The team found methane that appeared to be produced by microbes living inside the sediment itself, organisms so tough that they generate fuel from buried organic matter at crushing depths and near freezing temperatures. The community was sustained not just by chemicals seeping from the earth, but by bacteria actively making more fuel inside the rock, a self-sustaining chemical factory at the bottom of the world. Before this expedition, scientists knew chemosynthetic communities existed near hydrothermal vents in shallower parts of the deep ocean. Finding them here, at this depth, in multiple trenches, changed the scale of the story entirely. If chemical-eating ecosystems could exist in these two trenches, they could exist in the dozens of similar trenches scattered across the ocean floor worldwide. The lead scientists put it directly in their paper. These communities may be far more common than anyone previously imagined. We have been looking at the deep ocean and seeing almost nothing. The camera was aimed at the wrong thing. Life had found a completely different door and walked right through it. But here is the part that no one expected. Some of the creatures thriving in these communities are animals that break the basic rules of biology at the cellular level. The snailfish is one of them, and when you look at what this thing has done to its own body to survive down there, the word impossible starts to feel like it is not strong enough. Before we meet the snailfish, we have to understand the world it lives in, because the darkness in Challenger Deep is a different kind of darkness than anything most of us have experienced. Stand in a dark room at night. Some light leaks under the door, some filters through the curtains. Your eyes adjust and eventually you can make out shapes. That is still not true darkness. Close your eyes, put your hands over them, go into a windowless room and turn off the light. Even then, some researchers argue, a tiny amount of light enters from inside your own eyeball, from the chemical processes happening in your retina. At 6 miles below the Pacific Ocean surface, there is none of that. The sun's light reaches about 600 feet into the ocean before the water absorbs it completely. Below that, nothing. No light whatsoever. A depth of 6 miles is about 30 times deeper than sunlight can travel. Nearly every ecosystem on Earth runs on sunlight, even indirectly. A wolf eats a deer. The deer eats grass. The grass grew from sunlight. Go back far enough in any food chain, and you almost always hit a plant or an alga soaking up solar energy. The creatures at the bottom of the Mariana Trench have cut that connection entirely. Their energy source is chemistry. Specifically, it comes from reactions between toxic chemicals, mainly methane and hydrogen sulfide, and oxygen dissolved in the cold seawater. Bacteria living in these environments can harvest the tiny bit of energy released when these chemicals react. The bacteria grow. Other creatures eat the bacteria. The food chain builds upward from there, built on poison instead of sunlight. This process, chemosynthesis, was only discovered by science in 1977 when researchers stumbled onto the first hydrothermal vent community near the Galapagos Islands. Before that moment, biologists believed, with complete confidence, that sunlight was the foundation of all complex life. Every textbook said so, every model assumed it, the vents proved them wrong. And the Mariana Trench communities proved the lesson goes deeper, literally, than anyone realized. What makes the trench discovery especially significant is where the chemicals come from. The methane and hydrogen sulfide seep up through cracks in the ocean floor created by tectonic plates. The Pacific Plate is sliding underneath the Mariana Plate at this location, a process that cracks and buckles the rock, creating channels for superheated, chemical-rich fluids to escape upward. The Earth's own geological engine is feeding the ecosystem, and here is the part that stops scientists cold when they think about it too long. If life can run on chemistry instead of sunlight, if it only needs liquid water, chemical reactions, and time, then the requirements for life as we know them are far simpler than we thought. Simpler requirements mean more places in the universe where life could theoretically exist. Jupiter's moon Europa has a liquid water ocean beneath its icy surface. So does Enceladus, a moon of Saturn. Both are pitch dark. Both are cold. Both have no sunlight reaching their water. Both might have chemistry. The creatures 6 miles under the Pacific are living proof that darkness is not a deal breaker. and the animal that proves this point most dramatically is a pale, boneless-looking fish that scientists once thought was too fragile to survive even a short time at depth. Imagine a tadpole, make it bigger, maybe 7 or 8 inches long, give it see-through skin, remove the scales, then imagine it swimming through total darkness at 6 miles deep, completely at ease, while the pressure outside its body could flatten a steel drum. That is the snailfish. Scientists first described the Mariana snailfish as a species in 2014, after capturing specimens from extreme depths in the Mariana Trench. Since then, it has been documented at depths pushing nearly 5 miles, making it the deepest known vertebrate on Earth. A vertebrate means an animal with a spine, a backbone. The snailfish has one, sort of. The fish is small, pale, and almost translucent. Hold one up to a light and you could probably see its organs through its skin. It looks fragile. It looks like a creature that a strong current might bend in half. When scientists first saw footage of it gliding through the blackness at crushing depths, the word most researchers used was ghostly. But looks are almost everything with this fish. What it looks like and what it can actually do are two completely different things. The snailfish does not just survive down there, it thrives. It is the top predator in its ecosystem. It hunts amphipods, the shrimp-like scavengers that swarm in the deep trenches, and it does so with the casual confidence of a creature that knows it has no competitors. Nothing else its size lives at this depth. Nothing else even gets close. Scientists analysing its stomach contents found that the fish eats well, regularly. It is fat for a deep-sea creature, which tells you that food is available if you're built to find it. The community of tiny crustaceans that fill the trench floors actually gets more concentrated as depth increases. Amphipods gather in massive numbers near the bottom, and the snailfish, uniquely adapted to survive the pressure that kills everything else, has the whole buffet to itself. Here is what makes this remarkable from a biology standpoint. Every other fish species has a ceiling, a depth beyond which the pressure becomes too much. The snailfish keeps going past that ceiling, past the point where its closest relatives in shallower water would die, past the point where the rules of vertebrate biology say an animal with bones and proteins and cell membranes should be able to function. Scientists sequencing the snailfish's genome found something striking. The genes controlling the fish's basic cellular machinery, the instructions that tell its proteins how to fold, its enzymes how to function, its membranes how to stay flexible, all of them are different from those in shallow water fish. Specifically different. Adapted over millions of years. The snailfish's family of fish traces back to ancestors that lived near the ocean surface. About 20 million years ago, according to genetic analysis, the lineage that would become the Hadal snailfish began its descent. 20 million years of evolution, generation by generation, slowly rebuilding the molecular machinery to work under conditions that should make it impossible. And there is one more thing about this fish that no other creature at this depth seems to have managed. Its skeleton. Back in Chapter 2, we talked about the molecule called a piezolyte, the chemical cushion that keeps proteins from warping under extreme pressure. The snailfish has cracked this in a way that surprised researchers studying its genome. The molecule in question is called trimethylamine oxide. Scientists abbreviate it, but let us just call it the stabilizer. The stabilizer is a small compound that wedges itself around proteins inside cells and prevents them from changing shape when pressure squeezes in from outside. It works like a molecular bodyguard. Something tries to crush the protein, the stabilizer blocks it. In fish that live closer to the surface, the stabilizer is present in small amounts. As you look at fish species living deeper and deeper in the ocean, the amount of stabilizer in their tissues rises. Scientists discovered this pattern decades ago and assumed it would continue all the way to the bottom. The snailfish surprised them. Research published in the journal Cell studied the Mariana snailfish's biology in detail. One of the findings was that the stabilizer concentration does not keep rising indefinitely in the deepest fish. There seems to be a ceiling on how much of it a fish's biology can usefully carry. Scientists believe this may actually set a physical limit on how deep any fish can ever go. The snailfish is already near that limit. But the stabilizer is only one part of the pressure survival story. The snailfish's genome also reveals changes in the genes that control the fat in its cell membranes. Fats, in cold, high-pressure environments, tend to solidify. A solidified cell membrane cannot move, cannot transport nutrients, cannot do the basic work of keeping a cell alive. To get around this, the snailfish produces specific types of fats that stay fluid at these temperatures and pressures, fats that other fish simply do not make in these quantities. Additionally, the snailfish has expanded versions of genes related to handling a particular kind of cellular damage. Deep-sea environments, paradoxically, can cause a type of stress inside cells through chemical reactions involving oxygen. The snailfish's genome shows evidence of extra copies of genes that neutralize this damage, a kind of backup system layered on top of its regular biology. Each adaptation came from millions of years of selection. Snailfish that handled pressure slightly better survived slightly longer and had slightly more offspring. Repeat that for 20 million years. The result is an animal whose entire chemistry has been reorganized from the inside to match its environment. Think of a smartphone. The first version worked fine for phone calls, but every generation of update added features, removed bugs, and rebuilt the internal architecture to handle more. The snailfish is running software version 20 million years into its development. Its body is an engineering solution that evolution, through pure trial and error, arrived at over a timescale that makes human civilization look like a single afternoon. And yet the chemistry is only half the story. The physical structure of this fish is the part that made biologists genuinely stop and stare. Because the snailfish's bones are doing something that should not work. Your bones are hard for a reason. They support your weight, protect your organs, and give your muscles something solid to pull against. Hardness is the whole point. At 6 miles deep, hardness becomes a liability. Here is the problem. Bones are rigid. They hold their shape. But under the kind of pressure found at the bottom of the Mariana Trench, that rigidity creates stress points. Imagine crushing a dry cracker versus a piece of fresh bread. The cracker snaps. The bread compresses, bends, and springs back. Hard things shatter. Flexible things survive. The snailfish solved this problem by becoming more bread than cracker. Unlike the fish most people know, the snailfish has bones that never fully harden. Researchers examining specimens describe the skeleton as made of soft cartilage and incompletely ossified bone. Ossification is the process that turns a skeleton from soft, flexible cartilage into rigid bone. In humans, it happens during childhood. In most fish, it happens as they mature. In the snailfish, it never fully completes. The result is a body that can flex and compress slightly under pressure rather than cracking. The fish gives with the squeeze instead of fighting it. Scientists also found that the snailfish's skull is not a closed solid case the way most vertebrate skulls are. There are gaps, open spaces within the skull that allow it to accommodate the external pressure without fracturing. An open skull. In a fish, that works perfectly fine. There are no air-filled spaces inside the snailfish's body either, which matters enormously. Remember the styrofoam cup from Chapter 2 that was crushed to the size of a thimble? Air pockets compress catastrophically under pressure. Other deep-sea fish that go to moderate depth still have swim bladders, gas-filled structures for buoyancy. The snailfish has abandoned the swim bladder entirely. Its buoyancy comes from fatty, gel-like tissue in its body that happens to be very slightly less dense than water. Fat for ballast. No air, no rigid bones, no solid skull. The fish is essentially a soft, flexible, pressure-resistant container for the important biological machinery inside. Every rigid structure that might break has been replaced with something that bends. Some researchers have compared this design to the way engineers build submarines, not by making walls that resist pressure from outside, but by building structures where pressure is distributed evenly, so no single point takes a catastrophic load. The snailfish arrived at that same engineering solution. Evolution found it, without a drawing board, over millions of years. When scientists brought snailfish specimens to the surface, the fish deteriorated quickly. The low pressure of surface conditions is as alien to this animal as the high pressure of the trench would be to us. It literally falls apart above water. Its tissues soften further, its skin tears, and the animal dies within a very short time. Researchers now use special high-pressure tanks to study living specimens, replicating the trench environment in a laboratory. You can take the snailfish out of the trench, but you cannot take the trench out of the snailfish. And there is one more thing about this creature that scientists did not expect. In a world of absolute darkness 6 miles underwater, what is the point of having eyes at all? The snailfish still has eyes, small ones, reduced but functional. This confused scientists at first. What is the evolutionary logic of maintaining eyes in an environment with absolutely no light? Eyes are expensive to build and run. The brain dedicates real resources to processing visual information. If there is nothing to see, the animal should have lost its eyes millions of years ago. Many deep-sea creatures have done exactly that. The snailfish kept them, and when researchers sequenced its genome and studied the genes responsible for vision, the reason started to make sense. Normal fish living in shallow, sunlit water can see in multiple colours. They carry genes for several types of photoreceptor, the cells inside the eye that respond to different wavelengths of light: blue, green, red. Colour vision in bright conditions is useful. It helps fish identify ripe food, spot camouflaged prey, and recognise predators. The snailfish has lost almost all of those genes. The genes for colour vision are either gone entirely or barely expressed. What the snailfish has kept is a single functional gene for a photoreceptor that responds to very dim light, a gene called RH1. This receptor is tuned specifically for detecting low-level light with maximum sensitivity. In other words, the snailfish has stripped its visual system down to the one function that still matters in its environment: detecting faint, brief flashes of light in otherwise total darkness. Because darkness at the bottom of the ocean is not perfectly, permanently dark. Bioluminescence happens down there. Other animals produce flashes and pulses of cold chemical light made by living organisms, for all kinds of reasons we will get to shortly. A snailfish hunting for amphipods benefits from being able to detect those flashes. A sudden light means something nearby is alive. Alive often means edible. So the snailfish did not evolve blindness. It evolved hypersensitivity to the only light that exists in its world. This is a pattern that keeps appearing in deep-sea biology. Evolution does not simply remove things that are no longer needed. It repurposes them, strips them down to the one function they can still serve in an extreme environment, and optimizes that function to a degree that shallow water creatures cannot match. The snailfish's entire body is an example of this. Its skeleton, stripped down, its skull, opened up, its swim bladder, removed entirely, its colour vision, deleted, its pressure resistance chemistry, doubled down on. Every feature that would help a normal fish in a normal ocean has been either removed or rebuilt for the specific demands of the deepest trench on Earth. What is left is an animal that could survive nowhere else, but is perfectly suited to that one specific, extreme place. Researchers spreading across different trenches in the Pacific have found that the snailfish has colonised multiple trenches, spreading slowly across the ocean floor over the course of 1 million years. Each trench population has its own slightly different adaptations. The same basic design, pressure tested and rebuilt, applied to each unique environment. A species that spread across the planet by going deeper than anything else dared to go. But the snailfish is far from the only creature in these depths that should not exist. The next animal we meet is smaller, more numerous, and carrying something inside its body that humans put there. The amphipod is not glamorous. It looks like a shrimp that got stuck between two body plans, curved, segmented, a little translucent, usually an inch or two long in shallower water, but in the Hadal zone, some species grow to the size of your hand, an effect called abyssal gigantism that we will come back to later. Amphipods swarm the floors of the deepest trenches in massive numbers. They are scavengers, eating whatever falls from above, animal remains, dead fish, pieces of organic matter drifting down from the sunlit surface world thousands of feet overhead. They clean the trench floor the way blowflies clean a carcass. Efficient, relentless. This makes them critically important. Without scavengers like amphipods recycling organic matter at depth, nutrients would pile up uselessly at the bottom of the ocean and never re-enter the ecosystem. The amphipods eat the dead, and the snailfish eat the amphipods. The whole food web at this depth runs through them. Scientists started trapping and studying Hadal amphipods from multiple trenches around the Pacific in the 2010s. They collected samples from 6 different trenches, ranging from depths of about 4 miles down to nearly 7 miles. 90 animals total. More than 70% of them had plastic inside their digestive systems. Every single trench showed contamination. The deepest specimens, collected from nearly 7 miles down in the Mariana Trench, had the highest concentrations. Scientists found microplastic fibers, pieces smaller than 5 millimeters, in the hindguts of amphipods that had never been within thousands of feet of any human activity. Microplastics are pieces of plastic that break off larger items over time from UV exposure and physical stress. A plastic bottle floating in the ocean slowly shreds into smaller and smaller pieces. The smallest ones behave almost like food particles, drifting with ocean currents, sinking through the water column, eventually reaching the bottom. The Mariana Trench functions like a drain. Its funnel shape concentrates material falling from above. Plastics that spread across the Pacific eventually migrate toward the trench's axis. 1 litre of water collected from the trench contained as many as 2,000 individual microplastic pieces. And it is not just free-floating plastic in the water. When scientists looked at the deep-sea debris database, a collection of images and footage from thousands of dives over 30 years, they found that the deepest piece of identifiable trash ever recorded was a single-use plastic shopping bag, photographed 6 miles below the surface inside the Mariana Trench. A grocery bag, at the bottom of the world. The implications go beyond the trench itself. The amphipods carry this plastic inside them. The snailfish eat the amphipods. Plastic moves up the food chain, and it keeps moving. Studies of ocean food webs show that microplastics from the deep sea can travel upward through predator chains, eventually reaching fish that people eat. The trench is not separate from us. The trench is connected to everything, and right now, while we are 6 miles down looking at amphipods with plastic in their gut, there is something even stranger drifting down from above. Not fish, not plastic. Something that barely counts as matter at all. Look up from 6 miles underwater and you would see something falling. All around you, slowly, in every direction, tiny particles drift downward. Dead skin, fecal pellets, mucus strands, pieces of dead plankton, fragments of algae, clumps of bacteria, small bits of material from the surface world, falling in slow motion through miles of cold ocean water. Scientists call it marine snow. The name comes from how it looks in an underwater flashlight beam, a silent, endless white drift, particles swirling and tumbling as they sink. It takes weeks for marine snow to travel from the ocean surface down to the trench floor. The particles are slow, and as they fall, they clump together with other particles, growing slightly larger and heavier, picking up speed as they go. By the time they reach deep water, the flakes are sometimes a fraction of an inch across, dense little packages of compressed organic matter. Marine snow is the primary food supply for the majority of deep-sea creatures. The entire deep ocean, everything below the sunlit zone, runs on this slow, constant input of material from above. Sea cucumbers eat it off the sea floor, worms filter it from sediment, small crustaceans pick through it for nutrition. But here is the staggering math. Of all the food produced near the ocean surface through photosynthesis and other biological processes, less than 5% actually reaches the abyssal plain. More than 95% gets eaten or broken down during the long fall. Bacteria consume it. Small predators intercept it. Chemical processes break it down. By the time marine snow reaches the deepest floors, it is a very thin trickle of what started out above. An animal living on the abyssal plain is surviving on about 5% of the leftovers of a meal eaten far above by creatures that never think about the deep ocean at all. This food scarcity shapes everything about deep-sea biology. It explains why so many deep-sea animals are small. Smaller bodies burn fewer calories. It explains why many of them are slow. Slow movement burns less energy. It explains why some can go months or even years between meals and still survive. Evolution squeezed out every possible inefficiency and built bodies tuned to extract maximum value from minimum input. But occasionally something extraordinary happens. Researchers studying a deep-sea observation site off the California coast found that in certain years, massive blooms of algae form near the surface. The algae die and sink rapidly in huge pulses, delivering more food to the seafloor in a few weeks than would normally arrive over an entire year. Sometimes even decades worth of normal input arrives in a single event. For the animals down there, this is an event. They respond by moving more, feeding more, reproducing more, and sometimes the food that arrives is much larger than a snowflake. Sometimes a whale dies, and what happens next, in the days and years after a dead whale sinks 6 miles to the bottom, turns out to be one of the most complex ecological events on Earth. It involves creatures so specialized for this single moment in time that they exist nowhere else in the world. And one of them has no mouth at all. A lion can go about 2 weeks without food. A crocodile, if it drops its body temperature and stays still, can push that to a few months. These are impressive numbers for large warm-blooded or cold-blooded predators. The giant isopod, a deep-sea scavenger we will probably meet later, once went 5 years without eating in a Japanese aquarium. 5 years. It was not sick. It was not dying. It was simply waiting. The way a creature that evolved in a place where food might not come for years has learned to wait. Deep-sea animals have rewritten the rules of metabolism. Because when less than 5% of surface food reaches the bottom, and that trickle arrives unpredictably, the animals that survive are the ones whose biology can handle famine as a normal operating condition. Slow metabolism is one key. Deep-sea creatures burn calories at a fraction of the rate surface animals do. Their muscles are often built for low-speed endurance, not bursts of speed. Their hearts beat slowly. Their digestion moves slowly. Their growth is slow. Everything is dialed down to conserve the energy budget. Some deep-sea fish have such low metabolic rates that they can catch a single piece of food and run on it for months. Their digestive enzymes are specialized to extract every possible calorie from whatever they eat. Scientists studying the gut contents of deep-sea fish have found that some species eat just about anything that falls near them, including other fish, crustaceans, worms, and even pieces of plant matter that have drifted down from the surface. The trench environment also forces something unusual. When food does arrive in a large burst, such as a carcass or a bloom of marine snow, deep-sea animals respond by eating enormous amounts quickly and storing the energy as fat or glycogen. Their bodies can switch between feast and famine without the kind of cellular damage that would happen in a surface animal experiencing the same swings. Humans have a small version of this. Go a day without eating and you feel it. Your blood sugar drops, your focus blurs, your mood suffers. Surface fish starve and their organs start breaking down within days or weeks. The Hadal snailfish, the amphipods, the sea cucumbers crawling through the sediment below 6 miles of water, they have lived their entire evolutionary history in an environment where the next meal is never guaranteed. Their biology reflects every million years of that uncertainty. It is a kind of endurance that makes human athletic records look almost embarrassingly modest. And some of these animals have taken food finding to a place that seems less like biology and more like science fiction. The tube worm, for instance, does not wait for food to fall. It has permanently solved the hunger problem by cutting out the stomach entirely. 6 feet long, white tube, red plume waving from the open end like a feather in water. The giant tube worm looks like someone grew a creature in a laboratory as an experiment in how much biology you could remove before it stopped being an animal. It has no mouth, no stomach, no digestive system of any kind, no way to hunt, chew, swallow, or digest food in the way that almost every other animal on Earth does, and yet it grows to 6 feet long. It is one of the fastest growing invertebrates on the planet. In a world where most animals starve and move slowly, the tube worm does the opposite. Here is how it works. Inside the tube worm's body, packed into a specialized organ called a trophosome, live billions of bacteria. These bacteria are chemosynthetic, the same type of organism that forms the base of the trench food web. They take hydrogen sulfide from the hydrothermal vent water surrounding the tube worm and convert it into energy and organic compounds. The tube worm feeds them. The bacteria feed the tube worm. The worm draws in seawater through its red plume, absorbs the hydrogen sulfide and oxygen dissolved in the water, and delivers these chemicals to the bacteria living inside it. The bacteria process the chemicals, produce organic compounds, and the tube worm absorbs those compounds for nutrition. The two organisms have fused into a single system. This is called endosymbiosis, one organism living inside another, to the point where neither can survive without the other. The tube worm has not outsourced its digestive system. The tube worm has replaced its digestive system with a living organ made of other organisms. Scientists discovered tube worms in 1977 at the same hydrothermal vent site near the Galapagos Islands, where chemosynthesis was first documented. The discovery was so unexpected that the biologist who first saw footage of them reportedly refused to believe what she was looking at. 6-foot worms with no mouths, clustered around seafloor chimneys blasting toxic chemicals, in total darkness, thriving. The red plume colour is not decorative. It comes from hemoglobin, the same molecule that makes human blood red. Tube worms use hemoglobin in their plumes to carry sulfide and oxygen efficiently from the vent water to the bacteria inside, a transport system built from one of the most ancient biological molecules on Earth, repurposed for a completely alien chemistry. Their growth rate astonishes researchers. Under good conditions near active vents, tube worms can grow nearly a foot per year. In an environment where most creatures inch forward on survival's edge, the tube worm has found an energy source so rich and reliable that it can afford to grow fast. Vent communities built around tube worms support entire food webs. Crabs walk among the worm clusters. Small shrimp graze on bacteria growing on the worm tubes. Fish patrol the edges looking for anything slower than them. All of it built on a chemistry that runs without a single ray of sunlight. But the chemistry running this ecosystem comes from somewhere, from the Earth itself, and the source of that energy is even more violent and extreme than anything we have seen yet. Picture a chimney. Now put it on the ocean floor, make it black, jagged, about as tall as a house. Out of the top, blast water at 700 degrees Fahrenheit, so hot it would incinerate human tissue in a fraction of a second. Now surround that chimney with life. Hydrothermal vents form at cracks in the ocean floor, where tectonic plates are pulling apart or colliding. Seawater seeps down through the cracks, gets superheated by the magma below, picks up dissolved minerals and chemicals from the rock, and then blasts back upward through the seafloor. The temperature of the water coming out can exceed 700 degrees Fahrenheit. At sea level, that temperature would instantly vaporize water into steam. But at these depths, the immense pressure keeps the water liquid even at temperatures far above the normal boiling point. The hot water shoots up from the vent as a dark, mineral-rich plume. The minerals precipitate out of the water as it hits the colder surrounding ocean, building up the chimneys over time. Some of these chimneys are called black smokers because the mineral particles pouring out look like thick black smoke billowing from a factory stack. Right at the base of that 700-degree exit point, Within feet of water that would kill anything instantly, life crowds in. The gradient is everything. At the vent's centre, nothing survives. Feet away, where the scalding water mixes with the near-freezing surrounding ocean and cools to the range where chemistry happens productively, you find bacteria mats, tube worms, clams, shrimp, crabs. Some of the bacteria that live closest to the vents survive at temperatures above 200 degrees Fahrenheit. These are called thermophiles, heat lovers. They are some of the most extreme organisms on Earth, thriving in temperatures that would sterilize a kitchen. The shrimp near some vent communities have a unique adaptation, a light-sensitive organ on their backs that can detect the very faint infrared glow of the vent's heat, essentially a thermal sense organ, allowing them to navigate near vents without getting too close to the lethal centre. And the vents do not last forever. An individual vent can go active and shut down within years or decades as the underground plumbing shifts. When a vent dies, the community built around it dies too, or migrates. Scientists have found the ghost communities left behind, empty tube worm tubes standing in cold, dark water with no chemical supply, like empty city blocks after everyone has left. This raises a disturbing question. If vent communities need active vents to survive, and vents come and go, how do these communities spread? How do larvae from one vent find another vent miles away on the dark sea floor? Scientists are still working this out. The current thinking involves larvae being carried by deep ocean currents, riding the flow until they happen to land near a new chemical source. The odds are terrible, the distances are enormous, but the communities keep appearing, suggesting that some larvae make it. Life finds the cracks, literally, and in the darkness surrounding these vents something else is happening, something that makes the darkness almost irrelevant. Because in the deep ocean, animals have started making their own light. Before we get to bioluminescence broadly, meet the animal that uses it most theatrically. It is about a foot long, dark red, almost black. 8 arms connected by a web of skin, like a tiny cape stretched between them. 2 extra long filaments that extend beyond the web. Large blue-green eyes, which scientists measured as proportionally the largest eyes relative to body size of any animal on the planet. Its scientific name translates roughly to vampire squid from hell. Scientists did not name it that to be dramatic. They named it that in 1903 because the creature looked unlike anything they had seen, and the Latin name felt appropriate for something so deeply strange. The vampire squid is not a squid. It is not an octopus. It occupies its own separate order within cephalopods, sitting somewhere between the two groups, a living relative of an ancient lineage that branched off before modern squid and octopus fully diverged. Think of it as a window into a 200 million year old body plan that never fully modernized, one that survived by going somewhere nothing else followed. The deep mid-water zone, about 2,000 to 3,000 feet down. When threatened, the vampire squid performs one of the most spectacular defensive displays in the ocean. It flips its webbed arms up over its body, inverting itself, and holds the web high like a cloak being raised over a face. The interior of the web is lined with fleshy, spine-like structures. Inverted, the creature presents a spiky, bizarre shape that is almost nothing like what a predator would expect. If that fails, the vampire squid does something that no other squid or octopus can do. It ejects a cloud of glowing mucus from the tips of its arms, a thick, sticky, bioluminescent fluid that hangs in the water, lighting up with blue cold light while the squid escapes into the darkness. The cloud provides no camouflage. It is the opposite of camouflage. The goal is distraction, dazzle the predator, give it something glowing to stare at, vanish. The vampire squid survives in part because it lives in the oxygen minimum zone, a layer of the deep ocean where dissolved oxygen levels are so low that most predators cannot sustain themselves there. The vampire squid can, because of adaptations in its blood chemistry, that allow it to pull oxygen from water that would suffocate most other animals. It lives in the one zone where almost nothing else can follow it. Eating is another strange adaptation. Unlike most cephalopods that hunt actively, the vampire squid is a passive feeder. It extends those long filaments into the water and drags them, collecting particles of marine snow, fecal matter, dead cells, and organic debris. The vampire squid eats the drift of the ocean. It coats the particles in mucus and delivers them to its mouth. A cephalopod eating marine snow. Evolution found an angle no one predicted. The vampire squid survives on almost nothing because it burns almost nothing. Slow metabolism, extreme efficiency, a habitat no predator can easily reach, and it lights itself up in the dark. Speaking of which, the rest of the deep sea figured out bioluminescence a very long time ago, and what they did with it is much stranger than a defense cloud. Here is a number that stops people cold. About 75% of deep-sea animals produce their own light, 3 out of 4. In the largest habitat on Earth, the deep ocean, the dominant strategy for survival involves generating cold chemical light inside your own body. The deep ocean is not dark, it is full of light, living light, flashing and pulsing from nearly every direction. The chemistry behind bioluminescence involves 2 molecules. One is called luciferin, a light-producing compound. The other is called luciferase, an enzyme that reacts with luciferin and oxygen to produce a photon of light. No heat, no electricity. Just a cold chemical reaction that releases energy as visible light instead of heat. Animals use this light for several completely different purposes, and the variety of strategies is one of the most creative displays of evolution anywhere on the planet. Camouflage comes first. Some fish that live at mid-depths, not at the very bottom but still well below sunlight, face a danger from below. Predators looking up from greater depth can see their silhouettes against the very faint glow that filters down from the surface above. To eliminate this shadow, these fish produce bioluminescent light along their undersides at exactly the same brightness and colour as the light coming from above. They match the background, they disappear against the sky. This is called counter-illumination, and it is a trick that submarines inspired by it have experimented with to avoid detection. Communication is another use. Some deep-sea squid use complex patterns of bioluminescent flashing on their skin to signal to each other, possibly for mating or coordination. The patterns change rapidly, like a light display running across the surface of their bodies. Luring prey is a third strategy, and the anglerfish made this famous. Defense is a fourth strategy, as the vampire squid demonstrated. And then there is something even stranger, using bioluminescence as a searchlight for a type of prey detection that no other creature had thought of. One group of deep-sea dragonfish have evolved an organ that produces red light. Red light at depth is almost invisible to most deep-sea animals, because very few of them have receptors for it. The dragonfish points its red spotlight at prey and illuminates them without the prey being able to see the beam. To everything in the dragonfish's hunting ground, the environment looks dark. To the dragonfish, it is lit up. We will get into both the anglerfish and the dragonfish in the next 2 chapters, because they each deserve their own story. But consider what bioluminescence tells us about the deep ocean overall. 75% of animals lighting themselves up means that in the darkness of the deep sea, light is one of the most powerful tools available. It took evolution a few 100 million years to arrive at this conclusion, independently in dozens of completely different animal lineages. Fish, squid, jellyfish, crustaceans, worms, bacteria, all separately arrived at the same chemical solution to the same problem. The deep ocean is dark, but the creatures living in it refuse to stay that way. One of them built a lantern so sophisticated that it runs on a living organism, and it hangs the lantern directly from its own skull. The female anglerfish has a rod growing out of her skull. Not a horn, not a spine. A flexible appendage shaped like a modified fin ray, extending forward from the top of her head and curling down in front of her face. At the tip of that rod hangs a bulb of tissue. Inside the bulb live bacteria that produce bioluminescent light. The fish and the bacteria have a deal. The anglerfish provides the bacteria with nutrients and shelter inside the bulb. The bacteria provide the fish with light. The bacteria glow continuously, a cold blue-green pulse in the darkness. The anglerfish sits motionless in the water column with this lantern swinging in front of her mouth. Other creatures see the light. In the darkness of the deep ocean, light means food, or safety, or a mate. Smaller fish and crustaceans move toward it out of instinct. They approach. They get close. Then a mouth 3 times the width of the anglerfish's body opens. Rows of backward-pointing teeth snap shut, and the prey disappears. The anglerfish's mouth is large enough to swallow prey bigger than itself. Its stomach stretches. Its body expands. It can store enormous meals because in the deep ocean you eat when the opportunity comes, not when you feel like it. The teeth are another piece of engineering. They angle backward toward the throat. Once anything enters that mouth, moving forward is the only option. There is no reverse. Teeth pointing backward are not for chewing. They are for preventing escape. In smaller male anglerfish, an even stranger story unfolds. Males are tiny, sometimes 100 times smaller than females. They are born without a lure, without a developed digestive system, with a single biological mission: find a female. When a male locates a female through chemical signals in the water, he bites into her skin. His body then begins to fuse with hers. Blood vessels merge, organs connect. Eventually the male loses his independent existence and becomes a reproductive organ attached to the female's body. One female anglerfish can carry multiple males fused to her simultaneously. Scientists studying anglerfish reproduction describe this as one of the most extreme examples of sexual parasitism in the animal kingdom, though from an evolutionary standpoint it makes grim sense. In the vast, food-scarce darkness of the deep ocean, finding a mate is so unlikely that evolution selected for males who permanently attach the first time they succeed. The bioluminescent lure itself is still not fully understood. Scientists know the bacteria inside the bulb glow because of the same luciferin and luciferase reaction as other bioluminescent organisms. What they are still working out is how the anglerfish recruits these specific bacteria in the first place. The bulb appears to be open to the surrounding water, and researchers think the bacteria may be absorbed from the environment as the fish develops. The fish does not inherit glowing bacteria from its parents, it picks them up. A fishing rod made of bone, a living light bulb filled with bacteria, a stomach that stretches to any size, and somewhere in the darkness nearby, a weapon even more subtle than a glowing lure. A weapon that only the hunter can see. The anglerfish lures prey with visible light. It makes itself visible to trick things into coming closer. The dragonfish does something smarter. The stoplight loose-jaw dragonfish is a slender, dark-bodied fish roughly 6 to 12 inches long, found at depths from about 1,000 feet to 3,000 feet down. Its name comes partly from the red and green bioluminescent organs positioned around its face. It looks like a traffic light attached to a predator. Beneath each eye, the dragonfish carries a specialized organ called a photophore that produces red light. And this is where the biology becomes something that sounds more like a military advantage than a natural adaptation. Most deep-sea animals cannot see red light. Their photoreceptors are tuned to blue and green wavelengths, which travel better in deep ocean water. Red light at depth is essentially invisible to the standard deep-sea eye, because no red light exists at these depths naturally. No creature evolved to see it because there was nothing to see. The dragonfish evolved to both produce red light and to see red light. It carries photoreceptors in its eyes that can detect the wavelengths produced by its own chin organ. So when the dragonfish activates its red spotlight and sweeps it through the water column, the prey swimming around it see nothing unusual. Their world is dark. But to the dragonfish, the beam illuminates everything it touches. Small crustaceans, tiny fish, transparent organisms that would otherwise be nearly invisible. The The dragonfish hunts them under a private spotlight that only it can see. Imagine carrying a flashlight that broadcasts on a frequency no one else can detect. You can see everything around you. To everything around you, you are invisible. The dragonfish also has a secondary row of bioluminescent organs running down its belly and sides, producing blue-green light. Scientists believe this provides counter-illumination, matching the ambient light from above to eliminate the fish's silhouette for predators below. The dragonfish uses different light systems for different purposes simultaneously, running an invisible red spotlight while cancelling its own shadow with blue-green camouflage. The fish also has a very unusual jaw structure that gives it part of its name. The loose-jaw dragonfish can extend its lower jaw to an enormous angle, far beyond what any surface fish can manage, essentially unhinging its face to create a strike radius much larger than its body would suggest. Combined with the private red searchlight, this gives it a hunting system with almost no wasted movement. Locate prey you can see, but the prey cannot detect you seeing. Open jaw to maximum angle, strike. The dragonfish is invisible to its prey right up until it is too late. Scientists studying bioluminescence across the animal kingdom use the dragonfish as a key example of how light can be weaponized in ways that go far beyond a simple glow. The private red spotlight is an arms race that only one side knows it is running, and in the deep ocean, there are arms races everywhere you look. The next creature we meet solved the problem of predation by making its head completely see-through, so that nothing can sneak up on it from any direction, including from above. Imagine looking at a fish from the front and seeing right through its head. The barrelly fish has a dome of transparent tissue on top of its skull, filled with fluid, completely see-through. From the outside you can watch its brain sit quietly in the dark water. Through the dome, two vivid green tube-shaped eyes stare upward, like a pair of binoculars mounted inside a glass helmet. It is one of the strangest animals science has ever filmed in its natural environment, and for decades scientists did not even fully understand how its eyes worked. The barrelly lives at depths from about 2,000 to 2,600 feet, in the ocean's twilight zone where the last traces of surface light are still faintly present. The fish hangs nearly motionless in the water most of the time, its flat, wide fins barely moving, its tubular eyes aimed straight up toward the ceiling of the ocean. Scientists observing it from remotely operated vehicles describe it as patient, still, almost meditative. Here is what it is doing. It is watching. The green lenses on its eyes are tinted with a yellow pigment that works like a filter. Most deep-sea animals use bioluminescence to disguise themselves from below by matching the dim surface light. But the yellow pigment in the barrelized lenses blocks that glow and lets the fish distinguish between actual surface light and bioluminescent camouflage. Prey trying to hide by glowing at the right frequency cannot fool this fish. The filter peels back the disguise. For a long time, biologists thought the barrelly's eyes were fixed permanently upward, locked in place, giving the fish a permanent view of only what was directly above it. This seemed like a severe limitation. If the eyes could not move, how did the fish actually catch and eat anything? When prey drifted close overhead and the fish needed to lunge forward, how could it see what was in front of its own mouth? In 2008, researchers watching footage from the Monterey Bay Aquarium Research Institute discovered the answer. The eyes move. The entire barrel-shaped eye rotates inside the transparent dome, swiveling smoothly from pointing straight up to pointing straight forward. The fish watches overhead for the silhouette of a small creature against the faint glow above. When prey is close enough, the eyes rotate forward, the fish strikes, and it can see exactly what it is biting. No one had ever seen this because every barrelly specimen brought to the surface died before scientists could observe it. When the pressure changes as a fish ascends, the fluid-filled dome collapses, the eyes stop working, and the fish dies. Scientists studying dead specimens had frozen eyes, and no way to know those eyes had been mobile all along. It took live footage from a submarine, observing the fish in its actual environment to reveal the truth. Half a century of assuming the eyes were fixed. The fish had been rotating them the whole time. The barrelly likely hunts small animals like zooplankton, tiny crustaceans, and occasionally bits of jellyfish or siphonophores that drift through its feeding zone. Its transparent head probably also protects its eyes from the stinging tentacles of siphonophores, which the barrelly is observed swimming among frequently. A glass helmet protecting rotating eyes that see through camouflage. In the dark, patience is a weapon. Now scale up dramatically, because the next creature has been waiting down here for 160 million years, unchanged, carrying 14 legs and the patience to match. You have seen its tiny cousin, that little grey pill bug that curls into a ball when you pick it up from under a rock in the backyard. Now make it the size of a football. Give it 14 legs and a compound eye sensitive enough to see movement in near total darkness. Send it to the bottom of the deep ocean and leave it there for 160 million years. Meet the giant isopod. The largest confirmed giant isopods reach about 20 inches long and weigh around 4 pounds. Fossil evidence shows this body plan appeared roughly 160 million years ago, before the dinosaurs rose to their peak, and it has changed almost nothing since. The same 14 legs, the same segmented shell of overlapping armoured plates. When threatened, it curls into a perfect ball, tucking its legs inside the shell, turning itself into an armoured sphere that a predator cannot easily penetrate. Evolution looked at this design and decided, finished. Researchers studying giant isopods from different populations around the world have found that individuals across distant ocean basins are physically nearly identical. In most animal species, populations that are geographically separated for millions of years begin to diverge. Their genetics shift, their anatomy changes, new subspecies emerge. The giant isopod populations remain so similar that scientists think they either spread across oceans more recently than expected, or the deep sea environment exerts such a powerful, constant pressure that any variation gets eliminated. The ocean's depths are a strange kind of conservation machine. Extremity locks things in place. The giant isopod's survival strategy is built entirely around a two-part cycle. Eat enormously, then wait. When a whale carcass or a large dead fish sinks to the seafloor near it, the isopod eats until it physically cannot move. Its body swells with stored energy. It has been observed gorging until its legs fold beneath the weight. Then it retreats, slows its metabolism to a crawl, and sits on that stored energy for months, or longer. The most extreme documented case happened in a Japanese aquarium. A captive giant isopod refused all food for 5 years and 43 days before dying. 5 years, without a single meal. The animal's biology held its stored energy through all of it without any organ failure. Scientists believe this capacity is a direct product of deep-sea life. On the seafloor, food arrives in unpredictable bursts separated by long dry stretches. The animals that survived were the ones whose biology could outlast the gaps. Over millions of years, the giant isopod became one of the most metabolically efficient animals on Earth, capable of stretching a single meal across what would be a lifetime for most creatures. Its size is part of this system. Larger bodies store more energy per unit of metabolism. Bigger means you eat less often relative to what you store. The cold temperature of the deep ocean also slows the burn rate further, allowing even larger bodies to sustain themselves on limited food over long periods. Something even stranger appears when you look at a related group of deep-sea creatures: giant amphipods, giant sea spiders, giant squid. Across many unrelated animal groups in the deep ocean, species reach sizes far beyond their shallow water relatives. Scientists call this abyssal gigantism, and it is still not fully explained. Cold temperature, high oxygen in some zones, low predation pressure, and extended lifespans from low metabolism all contribute. The deep sea consistently makes things bigger, and stranger still, sometimes it builds creatures that are technically only one cell. Every living thing you've ever seen is made of cells, trillions of them, stacked together, organised into tissues and organs and systems. The smallest unit of life, a single cell, is something you need a microscope to see. Maybe 1/100th of a millimetre across, invisible to the naked eye. Now hold out your hand and picture something 4 inches across sitting in your palm, a disc of biological material, alive, one cell. The xenophyophore is the largest single-celled organism known on Earth. Scientists discovered these creatures on the deep ocean floor. They appear in multiple forms, some shaped like branching fans, some like irregular blobs, some like flattened plates. The largest ones reach about 4 inches across. A single individual is one continuous cell, its internal machinery contained in one membrane, without any division into separate compartments, the way multicellular organisms are structured. In 2011, a research expedition to the Mariana Trench photographed xenophyophores at a depth of 6.6 miles, the deepest any individual cell had ever been recorded on Earth. The previous depth record in a different trench had been nearly 2 miles shallower. These organisms are not just adapted to the deep ocean, they specifically thrive at the most extreme depths available. The xenophyophores name comes from Greek, meaning roughly bearer of foreign bodies, because of the way it constructs its structure. The cell builds itself using shells and fragments of other dead organisms found in the sediment around it, incorporating them into its body like a natural armour, assembled from graveyard material. It is the deep sea's answer to a hermit crab, built from the remains of whatever was around. What makes this creature ecologically important goes beyond the strangeness of its size. Research has shown that xenophyophores accumulate heavy metals in their bodies at levels that would poison almost any other organism. Lead, uranium, mercury. They absorb these metals from the sediment and hold them without dying. Scientists believe this makes them some of the most heavy metal resistant organisms on Earth, a property that could eventually have industrial or environmental applications. They're also, quietly, among the most important habitat engineers on the deep sea floor. Other animals shelter inside the complex branching structures of larger xenophyophores. Worms, brittle stars, small crustaceans, even fish use them. A study published in the journal Ecology found that deep-sea fish lay their eggs inside xenophyophore structures, using the single cell as a nursery. One creature, the size of a dinner plate, single-celled, providing shelter for the next generation of vertebrates. A living apartment building made of one cell. When that cell eventually dies, its structure collapses into the sediment and the nutrients cycle back into the seafloor system. Everything in the deep ocean feeds something else. Nothing is wasted. Even the death of a single cell matters. But few deaths matter more down here than the death of something much, much larger. A whale dies somewhere in the open Pacific Ocean. It weighs 40 tons. For a few days, the carcass floats at the surface, scavenged by sharks and seabirds. Then it starts to sink. The descent takes hours. The body falls through miles of cold, dark water, slowing as it goes, until it impacts the seafloor with a soft, heavy thud that disturbs the sediment in a cloud of grey. In the darkness, something detects it almost immediately. The first wave of visitors arrives within hours. Sleeper sharks, among the few large predators capable of reaching these depths. Hagfish, ancient jawless creatures that tie themselves in knots to strip flesh off bone. Giant isopods in numbers. They eat quickly and constantly, consuming what a human would consider an enormous amount of food, 130 pounds or more per day between all the scavengers combined. This stage lasts from a few months to a year and a half, depending on the size of the whale. When the flesh is gone and the clean skeleton lies on the seafloor, most people would assume the event is over. The food is gone, the visitors leave, life moves on. The whale fall has barely started. The bones of a large whale contain enormous amounts of lipids, the biological term for fats. Dense, calorie-rich fat locked inside the calcium structure of the skeleton. For the seafloor's ambient scavengers, fat locked in bone is inaccessible without a key. The key is one of the strangest animals in the ocean. Scientists studying the chemistry around whale skeletons on the seafloor have documented another shift. Sulfur-reducing bacteria colonize the bones themselves. They break down the fats chemically, releasing hydrogen sulfide in the process. The hydrogen sulfide then feeds a chemosynthetic community around the skeleton, similar to the communities around hydrothermal vents. A whale skeleton becomes a temporary vent. For up to 50 years, depending on the size of the whale, the skeleton sustains an entire ecosystem built around that one event, that one death. Researchers cataloguing the species present at documented whalefall sites have found nearly 200 species at a single skeleton, many of them found nowhere else in the ocean. For 50 years, a blue whale weighing 100 tons sinking to the seafloor becomes an island of life on an otherwise barren plain for half a century. Scientists call these structures stepping stones, points of biodiversity that allow species to spread across the deep sea floor by hopping from one whale fall to the next, across millions of years of ocean history. Whale falls may have shaped the evolution of deep sea species for as long as large whales have existed. The deep ocean's biodiversity may depend in part on whales dying in the right places at the right times, and one creature is so specialised for this event that it exists nowhere else. No mouth, no gut, no way to survive without a dead whale to drill into. The zombie worm was first documented in 2002 when a remotely operated vehicle spotted a cluster of them on a whale skeleton at the bottom of Monterey Canyon off the California coast, about 6,000 feet down. Scientists were not prepared for what they found. The worm, roughly 1 to 3 inches long, was growing directly out of whale bone, not lying on the surface, drilling through it. Root-like structures extended from the base of the worm's body into the calcium of the bone itself, reaching the lipids stored inside. The worm had no mouth, no stomach, no digestive tract of any kind. Yet it was alive, growing, and thriving. Its scientific name means bone devourer. Its common name, zombie worm, fits the image of a creature that feeds on death, reaching through bone with root structures instead of teeth, running on bacteria instead of digestion. Here is how it works. The root structures that penetrate the bone are not just physical anchors. They secrete a mild acid that helps dissolve the calcium structure and create access to the fat inside. Inside those roots live symbiotic bacteria, the same type of chemosynthetic partnership seen in tube worms. The bacteria break down the lipids and convert them into nutrients. The worm absorbs those nutrients through the roots. It eats through its roots, powered by bacteria living inside them, dissolving bone with acid, with no mouth at any stage of the process. Scientists later discovered that female zombie worms carry dozens of tiny parasitic males inside their own bodies. The males are microscopic, barely developed beyond the reproductive organs they need. In the deep ocean, finding a mate is so unlikely that males that locate a female and attach permanently survive longer than males that do not. Evolution selected for males that never let go. The females carry them as permanent passengers. The zombie worm is a key species in whale fall ecology. It unlocks the lipids from the bone and drives the chemosynthetic sulfur-producing community that keeps the whale fall alive for decades. Without zombie worms, whale bones would take much longer to process, and the sustained ecosystem they support might not form at all. Recent research has uncovered something alarming. Scientists placed whale bones on the seafloor at a deep-sea monitoring site off the Canadian coast and waited for zombie worms to appear. They waited 10 years. No zombie worms came, not one. Researchers suspect that expanding zones of low oxygen water in the Northeast Pacific, a pattern linked to warming oceans, are preventing zombie worm larvae from surviving long enough to settle. If the worm disappears from a region, the whale fall ecosystem chain breaks, and dozens of species lose their primary habitat. A creature with no mouth, no gut, and bones for a home is holding together one of the most important food webs in the deep ocean. And speaking of creatures that seem to break every rule about what animals are allowed to do, The next one has been doing it for 125 million years. In 1998, a Japanese remotely operated vehicle called Kaiko was descending through the Mariana Trench on a routine scientific survey. Its camera scanned the sea floor at 6.8 miles down. And there, floating quietly in the darkness, drifting on a slight deep ocean current, was a single-use plastic grocery bag. The footage sat in a scientific database for 20 years before researchers combing through archival deep-sea imagery spotted it. When they published their findings in 2018, the bag became the deepest piece of trash ever recorded on Earth. 6.8 miles below the surface, at the deepest accessible point on the planet. A grocery bag. It had arrived before any human ever reached that depth with their own eyes. Plastic does not just float. Larger pieces break down slowly from ultraviolet light and physical wear into microplastics, fragments smaller than 5 millimeters. These tiny particles are lightweight enough to drift with ocean currents, but they accumulate a coating of algae and bacteria over time, growing heavier and eventually sink. The Mariana Trench works like a drain on the bottom of the Pacific. Its funnel shape concentrates material falling from the water column. Currents channel debris toward the trench axis. Scientists measuring water samples directly from the trench found up to 2,000 microplastic pieces per litre. A litre is about 4 cups of water. 2,000 pieces of plastic in 4 cups of trench water. The physical presence is alarming enough. The biological impact is worse. Researchers sampling amphipods, the shrimp-like scavengers that swarm the deepest trenches, tested 90 individuals collected from 6 different trenches at depths ranging from 4 miles to nearly 7 miles. More than 70% of them had microplastic inside their digestive systems. Every single trench tested showed contamination. The Mariana Trench specimens showed the highest concentrations of all. These are animals that have never been anywhere near human civilization. They live at depths that crush submarines. The only way plastic reaches them is by falling from a surface world that these creatures have no contact with whatsoever. And it reaches them anyway. When Victor Vescovo piloted a submersible to the deepest point in Challenger Deep in 2019 and became the first person to reach that depth solo, one of the first things he saw through his porthole was what appeared to be a candy wrapper sitting on the sediment. Scientists sometimes struggle to convey the emotional weight of pollution findings using numbers and statistics. Vescovo's description did it in one sentence. The trench is not a separate world. It is the same world. Everything that enters the ocean eventually arrives at its lowest point. The creatures living 6 miles down have no vote on what falls through the water above them, and yet, somehow, the deeper we look, the more life we find down there. The next chapter goes further back in time than any of the creatures we have met, to an animal that barely changed while the dinosaurs rose and fell. Scientists described the goblin shark as a new species in 1898. The specimen that prompted the description came from Japan. When researchers first examined it, they were confused enough by its anatomy that for years they could not agree on how its jaw was even positioned in its skull. The jaw, as it turns out, was the whole story. The goblin shark has a long, flat, blade-like snout extending forward from its face. Beneath that snout hangs a lower jaw that appears retracted and almost calm when the animal is at rest. Then prey comes close. The jaw launches forward out of the mouth, extending dramatically in a slingshot motion, and snaps shut with rows of long nail-like teeth before retracting again. The launch is faster than any other jaw movement recorded in shark species. The jaw extends forward so dramatically that the animal's face momentarily changes shape, the snout remaining in place while the jaw shoots out below it. It looks less like a feeding event and more like the face is unfolding. The goblin shark's snout is covered with sensory organs called ampullae of Lorenzini, tiny pores that detect the minute electrical fields produced by living animals' muscles and nervous systems. All sharks have these. The goblin shark has an unusually high concentration of them along the entire length of its flattened snout, creating a wide detection surface. In the absolute darkness of deep water where eyes are nearly useless, this electrical sense becomes a precision tracking system. The shark drifts slowly, conserving energy, its low-density flesh making it nearly neutrally buoyant so it can hover without swimming. The electrical sense guides it. When the signal gets strong enough, the jaw fires. Scientists call the goblin shark a living fossil. That phrase sometimes gets overused, but in this case the lineage genuinely traces back 125 million years through the fossil record to ancestors that swam in Cretaceous seas while non-avian dinosaurs walked on land. The goblin shark's family has one surviving species. One. Every other branch of its lineage died out over the last 100 million years. The goblin shark itself persisted, unchanged in its basic design, going deeper into the ocean as the world above changed beyond recognition. It is pink, almost translucent, because its skin is thin and the blood vessels underneath show through. Scientists have observed that as goblin sharks age, their bodies actually shorten slightly as the flexible cartilage of their skeletons compresses under years of pressure. An old goblin shark is slightly shorter than a young one. They are rarely encountered. Most known specimens came from deep-sea fishing nets that scooped them up by accident. A goblin shark that ended up in a Tokyo aquarium survived only a week before the conditions killed it. What we know about this animal's biology comes mostly from dead specimens and a handful of brief video clips. 125 million years of survival. And we still barely know how it lives. Something is about to get stranger than a jaw that fires like a slingshot, though. The next creature solved the gigantism problem by not bothering with being giant at all, and solved the problem of looking threatening by looking like a Disney character instead. In 2019, a remotely operated vehicle descending into the Java Trench in the Indian Ocean was filming the seafloor at about 4.5 miles down when a small pale shape appeared on the camera. It was about 8 inches long, gelatinous, with 2 fins extending above its eyes like ears. It flapped those fins slowly and hovered a few inches above the seafloor, almost weightless, drifting forward in tiny pulses. The Dumbo octopus, named for the Disney elephant that could fly using its enormous ears, had just set a new record as the deepest cephalopod ever documented on film. 4.5 miles, and it looked completely relaxed about it. Scientists confirmed the sighting in a study published in 2020. It remains the deepest recorded observation of any octopus species by a significant margin. The Dumbo octopus exists as about 18 different species in the same genus. They live across all the world's oceans from about 3,000 feet down to nearly 5 miles deep, in temperatures around freezing, in total darkness, under hundreds of atmospheres of pressure, and they manage all of this while looking, by most accounts, thoroughly unbothered. The Dumbo octopus abandons several tools that its shallow water relatives rely on. It carries no ink sac because releasing ink in the deep ocean would serve no purpose since there is no light for it to block. It cannot change colour for camouflage the way shallow water octopuses do. It gave up jet propulsion as its primary movement style. Instead, it relies on those ear-like fins, flapping them in slow, rhythmic beats to propel itself along the seafloor or through the water column, conserving maximum energy in an environment where burning extra calories carries real cost. Most octopuses have a defined hunting pattern. Locate prey, pursue it, grab it with arms, manipulate it, eat it in pieces. The Dumbo octopus does something different. It finds worms, small crustaceans, and other slow-moving prey on or near the seafloor, and swallows them whole. One motion, no pursuit, no manipulation, no wasted energy in subduing prey that is unlikely to escape at depth anyway. The arm web connecting all 8 arms forms an umbrella shape. When spread wide, it slows the animal's descent like a parachute. When pulled in, the animal streamlines for faster movement. Combined with the fin flapping and a siphon for emergency propulsion, the Dumbo octopus has 3 different movement modes for 3 different situations. Female Dumbo octopuses reproduce in a way unique among their relatives. They carry eggs with them at all stages, attaching them to hard surfaces like rocks and coral when the time comes. Scientists think this may include the ability to store sperm for extended periods, allowing a female who has only encountered a male once to fertilize eggs later when conditions are right. The only cephalopod confirmed to live in the Hadal zone, doing it with ear fins, no ink, and the movement efficiency of a creature that figured out the math of survival at depth better than most anything else down there. But the world it swims in was not always this shape, and will not always remain it. The trench itself is still being built, by forces operating on a scale that makes even the giant creatures of the deep look small. The Mariana Trench was not dug, it was bent. Here is what happened in slow motion over 50 million years. The Pacific Ocean floor is a tectonic plate, a slab of rock about 60 miles thick, stretching across most of the Pacific. Tectonic plates move. They carry ocean floor and continents like cargo on very slow conveyor belts. The Pacific plate moves roughly a few inches per year in a direction that brings its western edge into contact with a much smaller plate called the Mariana Plate, which sits just to the west. When two oceanic plates collide and one is denser and colder than the other, something has to give. The denser plate bends downward and slides beneath the lighter one. This is called subduction. The Pacific Plate, carrying some of the oldest and coldest oceanic crust on Earth, roughly 180 million years old, is so dense that it bends at a steep angle as it pushes under the Mariana Plate. That bend in the Pacific Plate, where it angles sharply down into the Earth's interior, is the Mariana Trench. The trench is the surface expression of a plate folding itself toward the Earth's mantle. As the Pacific Plate descends, it carries water locked in its rock down with it. Under heat and pressure, that water gets released into the overlying mantle, lowering the melting point of the rock above. The rock melts partially and rises as magma, eventually erupting through the seafloor to the west as a chain of underwater volcanoes. Those volcanoes, built up over millions of years of eruptions, become the Mariana Islands. The trench and the island chain above it are two products of the same process, one pulling down, one pushing up, driven by the same plate collision happening at 3 to 4 miles per year. The subduction zone also generates earthquakes. When the Pacific Plate grinds against and beneath the Mariana Plate, stress builds and releases in seismic events. Some of these earthquakes are enormous. Scientists studying the seafloor have found evidence of regular major seismic activity along the trench. The snailfish's adapted short lifespan may be a direct response to this instability. A trench floor that shakes regularly is a trench floor that can suddenly bury everything living on it under tons of shifted sediment. The trench has been actively deepening for tens of millions of years and will continue to do so as long as the plate collision persists. The Pacific Plate has not finished its journey. It will continue bending downward into the mantle, feeding the subduction zone, rebuilding the trench's geometry year by year. This geological engine, the one building the deepest place on Earth, also built the conditions that make chemosynthetic life possible. The cracks in the rock created by the subduction process provide pathways for methane and hydrogen sulfide to seep upward from deep inside the Earth. The creatures thriving at the bottom of the trench are living off the chemistry, released by the same forces bending the planet's crust beneath their feet. The trench is alive with geology. The biology could not exist without it. And the next story is about the moment humans decided they needed to see it for themselves. On January 23, 1960, two men climbed into a metal sphere and sank toward the deepest point on Earth. Don Walsh, a Navy lieutenant, and Jacques Picard, a Swiss engineer, descended in a vessel called Trieste. The descent took nearly 5 hours. The sphere they sat in was thick steel, designed to resist pressure that would crush ordinary materials to fragments. When they reached the bottom of Challenger Deep at about 35,000 feet, they had 2 windows through which to observe the deepest place humans had ever visited. One of those windows developed a crack during the descent. They stayed for 20 minutes. They saw sediment, a flatfish, or what appeared to be one, moving along the bottom, proof that vertebrate life existed at the deepest point in the ocean. Then they released their ballast and rose back to the surface over 3 hours. The next human visit to Challenger Deep came 52 years later. James Cameron, the filmmaker behind Titanic and Avatar, funded the construction of a custom deep-sea submersible and piloted it solo to the bottom in 2012. He spent about 3 hours on the seafloor, photographing and filming. He reported the bottom as a flat grey plain of fine sediment. Lunar, he said. He saw organisms less than an inch long. He described the scale as humbling in a way that space footage does not fully capture. Then Victor Vescovo, a retired naval officer and investor, built a more advanced submersible and in 2019 made the deepest dive ever recorded, pushing slightly deeper than Cameron's mark. Over the following years, he dove Challenger Deep a total of 15 times, more than any person in history. Scientists accompanying him on later dives discovered new species, collected previously unreachable samples, and mapped the floor of the trench with a resolution that revealed details no previous survey could. In total, as of now, fewer than 30 people have ever visited the Challenger Deep. More humans have walked on the surface of the Moon than have been to the deepest point in the ocean. The Moon is 239,000 miles away. Challenger Deep is less than 7 miles straight down. Distance is not the barrier here. Pressure is. And the engineering challenge of surviving that pressure while gathering useful scientific data is still one of the hardest problems in exploration. The submersibles that work at these depths represent some of the most precisely engineered machines ever built. Even they are tested to the absolute limit by what the trench does to everything that enters it, because some machines have not come back. In May 2014, a remotely operated vehicle named Nereus was 6 miles underwater in the Kermadec Trench, northeast of New Zealand, working through a planned 9-hour dive. 7 hours in, the research ship above lost contact with the vehicle. No warning, no signal. The communication simply stopped. The crew deployed search equipment. Eventually they spotted debris floating on the surface, pieces of the vehicle. The pressure at 6 miles had found a weak point, and the implosion happened in a fraction of a second. Implosion, rather than explosion, is what pressure failure looks like at depth. When a sealed structure fails at 16,000 pounds per square inch, the surrounding water does not push it outward like a bomb. It collapses inward, the external pressure so vastly overwhelming the internal pressure that the structure folds in on itself catastrophically. A styrofoam coffee cup crushed to the size of a thimble takes weeks of gentle, constant pressure. Nereus took a fraction of a second. Nereus was built by Woods Hole Oceanographic Institution, one of the premier ocean research organizations in the world. It was the only scientific vehicle available to the global scientific research community that could work in water deeper than 4 miles. Its loss was described by researchers as a devastating blow to deep ocean science. 5 planned expeditions had to be cancelled. The engineering challenge of building machines for these depths is unlike almost any other design problem. Every sealed compartment is a potential failure point. Electronics must be kept at atmospheric pressure inside sealed housings, while the housing itself resists 16,000 pounds per square inch from outside. Glass, ceramic, titanium, and syntactic foam, a material made of hollow glass beads suspended in resin, are the materials that survive best. Standard metal housings implode. Even the materials that work require cycling through extreme pressure changes repeatedly. Every dive and ascent stresses the structure differently. Small cracks can develop. Over time, they grow. Nereus had made many successful dives before its final one. The failure, investigators concluded, was likely stress fatigue, repeated pressure cycling that eventually reached the material's limit. Scientists working in the field after the Nereus loss pointed out that the deep ocean is so physically demanding on equipment that it takes longer to develop reliable tools for exploring it than it took to develop the tools for exploring space. Space is vast and cold and lethal, but the structural engineering challenge of a vacuum is simpler than the engineering challenge of 16,000 pounds per square inch of active, corrosive, constantly pressurized salt water. We have been exploring space with telescopes and satellites and crewed vehicles for decades. Our deepest ocean trenches remain largely unmapped because the tools to explore them are so difficult to build and so easy to lose. And the scale of what we still do not know is even more striking than the machines we have lost. Scientists estimate that humans have explored and visually mapped less than 1/10 of 1% of the deep sea floor. Read that again. We have seen less than 1/10 of 1% of the ocean floor below about 3,000 feet. Not mapped at low resolution. Seen, up close, in enough detail to actually know what is there. The deep sea covers more than half of Earth's total surface. It is the largest habitat on the planet, and we have direct, detailed knowledge of a sliver so small it barely registers as a fraction. Compare it to this. We have complete high-resolution maps of the Moon's surface. We have detailed maps of Mars. We have mapped asteroids, Venus, Jupiter's moons. These objects are millions of miles away. The bottom of our own ocean is, at its deepest, 7 miles below the ship floating above it. We know the surface of Mars better than we know Earth's ocean floor. The reason is a combination of pressure, darkness, cost, and equipment loss. Satellites map land surfaces by bouncing signals off them. That technique does not work through miles of ocean. Sound-based mapping can create rough outlines of the seafloor's topography, but seeing it, collecting samples from it, observing the creatures living on it, requires equipment that is enormously expensive to build, extraordinarily difficult to operate, and, as Nereus demonstrated, possible to lose in an instant. Every time a new remotely operated vehicle reaches a previously unexplored section of the deep sea floor, it finds things that were not expected. New species, new geological features, new chemical processes. The Fenduji expedition that found the deepest chemosynthetic communities in the Mariana and adjacent trenches was not looking for them in those specific locations. The xenophyophorus photographed in the Mariana Trench had not been documented there before. The snailfish was formally described only in 2014. In a survey of the Clarion-Clipperton Zone, a region of the Pacific seafloor at about 13,000 feet depth that mining companies have interest in, scientists catalogued nearly 800 species of animals in the sediment. The majority of them had never been described before. The majority of them were, by scientific definition, unknown to humans prior to that survey. Every expedition finds new life. Every sample contains organisms no one has named. Every camera pointed at a new section of seafloor shows something unexpected. Scientists estimate the deep sea likely contains millions of species that have never been documented. Not hundreds, not thousands, millions. Most of them invisible to us not because they are rare, but because we have never gone to where they live. The ocean has been here for billions of years. We have been seriously exploring it for a few decades. The gap between what exists and what we know is one of the most staggering margins in all of science. And something is threatening this unexplored world before we ever get the chance to understand it. Scientists looking for life on other planets face a fundamental challenge. They do not know exactly what they are looking for. The assumption for most of the 20th century was that life requires sunlight. A planet habitable for life needed to be in the right distance from its star, close enough for liquid water, far enough not to cook. This zone is called the habitable zone, and for decades astronomers searched for planets in it. Then came the hydrothermal vents in 1977, then the tube worms and chemosynthetic communities, then the snailfish at 6 miles deep eating amphipods in total darkness. Then the worms and mollusks discovered in the Mariana and adjacent trenches, thriving in a world with no sunlight, no photosynthesis, no connection to the surface at all. The definition of habitability changed. If life can run on chemistry instead of sunlight, if liquid water and chemical gradients are sufficient, then the list of potentially habitable environments in our solar system becomes dramatically longer. Scientists studying this problem now focus on 2 moons in particular. Jupiter's moon, Europa, has a liquid saltwater ocean beneath a crust of ice, possibly 60 miles deep. Its surface receives almost no usable sunlight. Scientists believe the ocean floor of Europa may have hydrothermal vents, driven by tidal heating from Jupiter's enormous gravitational pull, flexing the Moon's interior. The chemistry could be similar to Earth's vent communities. The temperature and pressure ranges overlap. Saturn's moon Enceladus has similar properties. It actively ejects plumes of water vapour from its southern pole, which the Cassini spacecraft flew through and analysed. Those plumes contain water, organic molecules, hydrogen, and silica particles that suggest hydrothermal activity at the seafloor. Enceladus ticks nearly every box that deep-sea chemosynthetic communities need. NASA's Europa Clipper mission, launched in 2024 and scheduled to arrive in the Jupiter system in 2030, will study Europa's ocean chemistry from orbit. Scientists designing the mission have spent years studying the creatures of Earth's deep ocean as the closest analogue available for what they might find. The snailfish is a model for pressure adaptation. The tube worm is a model for chemosynthetic life systems. The xenophyophore raises questions about the size limits of single-celled life. The amphipod, carrying microplastics in its gut at 6 miles depth, reminds scientists that surface chemistry spreads to the deepest possible environments over time, with or without anyone carrying it there deliberately. What these creatures prove, collectively, is that life does not need what we thought it needed. It needs liquid water. It needs a source of chemical energy. It needs enough time. That combination is far more common in the universe than a planet sitting at the right distance from a star. The deep ocean is not just an interesting place on Earth. For scientists thinking about life elsewhere, it is the best evidence available that other places are worth looking. But evidence is only useful if what it describes is still there to study. Scattered across the deep ocean floor, particularly on the abyssal plains at depths of about 13,000 feet, are billions of dark, rounded lumps about the size of a potato. These lumps, called polymetallic nodules, have been slowly forming on the seafloor for millions of years, accreting layer by layer around a tiny seed particle. Each nodule is packed with manganese, cobalt, nickel, copper, and rare earth elements. The same materials that go into electric vehicle batteries, smartphone screens, renewable energy components, and advanced electronics. The demand for these materials is rising globally. The nodule fields on the deep Pacific floor contain concentrations of these metals that dwarf most land-based deposits, and companies have been submitting proposals for years to mine them. In 2022, an industrial trial of a commercial mining machine operated at 13,000 feet depth on the eastern Pacific floor, collecting more than 3,000 tons of nodules in a test run. Scientists who surveyed the area before and after the test found that the abundance of animals in the disturbed seafloor tracts dropped by 37%. Species diversity dropped by 32%. A published review in 2025 examined data going back 40 years to the first test mining experiments. It found that biological impacts from nodule removal persist for decades. Some species began returning after 4 decades, others had not. Decades to partially recover from a test. Hydrothermal vent communities face an additional threat. The mineral deposits formed at vents, called seafloor massive sulfides, are also rich targets. But scientists who reviewed the evidence concluded that mining these deposits would likely cause permanent biodiversity loss. The vent communities are so tightly connected to the active chemistry of the vent that removing the mineral structure disrupts the entire food web. Species found nowhere else in the ocean, creatures that evolved over millions of years to survive in that specific chemical environment, would lose their only habitat. Here is the problem that scientists keep raising. We have seen less than 0.1% of the deep sea floor. The survey of the Clarion-Clipperton zone, where most of the nodule mining interest is focused, found hundreds of previously unknown species. The areas proposed for mining are not empty. They are full of life we have never documented, following processes we do not yet understand. Mining proposals are moving forward. International regulatory frameworks for deep-sea mining remain incomplete. The International Seabed Authority, which oversees mining rights in international waters, has been working on rules for years without finalising them. Companies are already holding exploration licenses. The question researchers keep returning to is one of timing. When we finally do map the deep sea floor in detail, when we finally do understand what is living there and how its ecosystems function, we may discover we have already destroyed significant parts of it. The window for understanding the deep ocean may be closing before the cameras get there. The creatures in this video have survived pressure, darkness, starvation, and geological upheaval for millions of years. The thing that might finally end them is a machine dragging a rake across a seafloor we barely know exists. The Mariana Trench is the most famous deep-sea trench on Earth. It is the deepest, the most studied, the most visited. It is one of roughly 46 major ocean trenches worldwide. Most of them have never been visited by any manned or remotely operated vehicle. Several have been photographed only with simple drop cameras. Some have barely been acoustically mapped. The species living in them, the chemical processes operating in them, the geological activity occurring within them remain almost entirely unknown. Consider what the Fenduji expedition found in just one series of dives into the Kuril-Kamchatka Trench and the Western Aleutian Trench, two trenches that had received far less attention than the Mariana. Researchers encountered the deepest chemosynthetic communities ever documented. Communities that, by the science team's own assessment, challenged the current models for how carbon cycles through the deep ocean. The discovery was not expected. They found it because they went and looked. The Tonga Trench in the South Pacific reaches nearly 7 miles. The Philippine Trench runs nearly 6 miles at its deepest. The Kermadec Trench, where Nereus imploded, goes down to more than 6 miles. Each of these trenches has its own geological character, its own chemical environment, its own set of species adapted to exactly that specific place. A creature found in the Mariana Trench may not exist in the Kermadec Trench. A vent community in the Philippine Trench may have species that appear nowhere else. Scientists studying the snailfish found that different trench populations have different genetic variations, shaped by their specific local environments over millions of years. Each trench is its own evolutionary laboratory, running its own experiment in extreme adaptation. What we found in the Mariana Trench, specifically the Mariana Trench, is already rewriting biology textbooks. The science team that published the Chemosynthetic Community Discovery in Nature stated directly that similar communities may be far more widespread than previously assumed. If every trench system in the world has its own version of what Fenduji found in the northwestern Pacific, the deep-sea biosphere is several orders of magnitude richer than any existing estimate. And essentially none of it has been seen. The deep ocean is not a mystery the way a math problem is a mystery, something to solve if only someone would sit down and work it out. It is a mystery the way a continent is a mystery before anyone crosses it. The territory is real, the features are real, the organisms are real. They have been here for tens of millions of years. They will persist until something removes them. We've arrived at the edge of this continent late. The cameras are just reaching the shore, and every minute of footage from those cameras confirms the same conclusion, one that changes how we understand life on Earth, and perhaps life anywhere. Go back to the beginning, 6 miles down, total darkness, pressure 1,000 times greater than the air on your skin, temperature near freezing, no sunlight, no photosynthesis, no connection to the world you live in, and thousands of animals thriving, a fish with no solid bones ruling the food web, single cells the size of dinner plates sitting on the sediment, worms with no mouths drilling through whale bones with acid and bacteria, shrimp with microplastics in their guts from a surface world they have never visited, a grocery bag drifting past a living community that was building ecosystems before the first humans walked out of Africa. All of this has been here the entire time. We simply had not gone to look. The discoveries in the deep ocean over the last few decades have changed biology in ways that researchers are still mapping. Chemosynthesis as a foundation for entire ecosystems rewrites the requirements for life. Pressure adaptations in the snailfish demonstrate that vertebrate biology is more flexible than anyone predicted. Bioluminescence in 75% of deep-sea species rewrites the nature of a dark environment. Whale falls sustaining biodiversity for 50 years rewrites nutrient cycling in the ocean. Each of these findings matters beyond the deep ocean itself. They shift how scientists think about what life can do, what it requires, where it might be found. The creatures in the deep ocean did not adapt to their environment as a secondary option. They got there first. Chemosynthesis predates photosynthesis as a biological strategy. Life at extreme depth, at extreme pressure, at extreme chemical environments may represent the original template for Earth's biology. The surface world, sunlit and temperate, may be the exception, a pleasant suburb built long after the original city was already running smoothly underground. That is still debated. But the evidence from the deep suggests the range of what life can do is far wider than what we observe on the surface. It also means that the window to understand this world is one we have barely opened. Every expedition to a new section of the deep sea floor finds new species. Every sample of sediment contains biology that has not been named. The deep ocean, covering more than half of Earth's surface, remains the largest unexplored environment on the planet, filling rapidly with human debris while science scrambles to document it before that debris and the mining machines alter it permanently. The fish with no skull, the worm with no mouth, the single cell the size of a plate, the octopus with ear fins flying through the deepest water on Earth. These are not curiosities. They are evidence. Evidence that life is more resilient, more creative, and more widespread than any previous generation of scientists dared to assume. The rules of biology everyone learned in school were written before we went deep enough to see what biology actually does when the pressure is on. Down there, it does something extraordinary, and we have seen less than 1/10 of 1% of it.